Ladies and gentlemen, thanks for joining GenScript webinar today. I'm your host, Boon Hua. GenScript is the world's leading technology and service provider of life science research, development, and manufacturing. Driven by the corporate missions of making people and nature healthier through biotechnology, GenScript provides premium, convenient, and reliable services and products that speed up the evolutions of whole biotech and biopharma industry. In today's webinar, we are going to discuss the solutions of new COVID-19 variant and post-vaccination monitoring. Today, we have invited Dr. Chengxun Su, field application scientist from GenScript, to discuss about this topic. Dr. Chengxun has extensive experience in developing bioengineered platforms for disease modeling and drug testing applications, where she applied cutting-edge technologies in cell biology, microfabrications, microfluidics, and molecular biology to develop novel tools to support fundamental and translational research. Dr. Chengxun also spearheaded several collaborations with clinicians to investigate immune dysfunction and inflammatory profile in patients who recover from COVID-19, paving the way for potential intervention strategies. So before we get started, here are some of the introductions on the interface that you are using now. At the center area is the main presentations where Dr. Chengxun will be sharing her slides. At the top left corner, there's the speaker bio where you can check the details of our speaker today. At the bottom left is a Q&A box. Feel free to ask any questions throughout the presentations and we will answer it after the presentations. At your top right corner is the related contents of our product, so feel free to download it and check it out later. And at the bottom right is the survey that we provided. Please help us to answer it, and it helps us to improve the service and products that we have. So without further ado, I would like to pass the stage to Dr. Chengxun. Over to you.
Thank you, Boon Hua. So good afternoon, everyone. So thank you very much for tuning in today for today's webinar. So for today's webinar, I will first give an introduction on the Omicron variant and also protective immunity and neutralizing antibodies. Secondly, I will introduce the surrogate virus neutralization test, which is abbreviated as SVNT. And this is also the technology behind the CPAS kit. So for this section, I will discuss neutralizing antibody as the correlate of protection, and also how can we detect neutralizing antibody against a variant of concerns using SVNT. So thirdly, I will discuss the clinical significance of evaluating the neutralizing antibody response, including how can the SVNT be used for the next generation vaccine development, and how is the neutralizing antibody dynamics and durability post-infection or vaccination. And lastly, I will also discuss the possibility of neutralizing antibody-guided booster dose regimen. So last but not least, I will also go through the tools for your Omicron research. That means the reagents and services that GenScript provides for Omicron research. So firstly, I would like to give an introduction on the most recent variant of concern, Omicron. So Omicron is firstly detected in November 2021 in South Africa. So currently it has spread to more than 110 countries at the end of last year. So the Omicron variant is reported to have a much higher transmissibility. So as we can see from the new reported case graph here, it has caused the global caseload to new highs, which is close to 3 million cases. And also it is displacing Delta in some countries. So as shown by this graph here, so the different colors represent different variants of concerns. In South Africa, we saw that before the onset of Omicron, Delta is the main variant of concern. And ever since December, it has become the main variant circulating. And this is the same trend for USA right now. So besides these two countries, we also observe this consistent trend of Omicron being the dominating variant of concern in most other countries. So this very increased transmissibility of the Omicron can be ascribed due to its the large number of mutations present on the spike protein. So on the on its spike protein, there are more than 30 changes to the spike protein itself, and 15 of them is on the receptor binding domain region, which is the region that mainly interact with human cells. And besides this, there are also 2 mutations on the nucleocapsid protein. So in the most recent WHO meeting on how can we cope with this new Omicron variant, 3 key research priorities were identified. The first priority is to identify the ability of post-vaccination and correlation sera to neutralize Omicron. So this can be identified using neutralization tests, such as the conventional virus neutralization test, and also the SVNT, which we are going to introduce later on. And then the second priority is to identify the ability of current vaccines to prevent a mild or severe disease. So this study can be, such insights will be able to derive from the real-world vaccine protection data. And then lastly, it is also very important to evaluate the ability of current therapeutic approaches for treatment of COVID-19. So next, I'm going to introduce how is the mechanism of infection for the SARS-CoV-2 virus. So as shown here, when the virus comes into contact with the cell, the spike protein on the surface of the virus will interact with the ACE2 receptor that is expressed on the human cells. So once the spike protein and ACE2 receptor come into contact, it will trigger the internalization of the virus into the whole cell. So when we take a closer look at the protein-protein interaction region, we understand that it is the S1 RBD region, which is the receptor binding domain region of the spike protein that specifically binds to the ACE2 receptor. So when our body receives vaccination or when we are infected, we will develop adaptive immunity, which will protect us from future infection. Adaptive immunity consists of 2 arms. The first arm is mainly mediated by T cells. So T cells is usually activated after the virus enters the whole cell, and then it will kill the infected cells and to stop the viral replication. And hence T cells is very important, plays a very important role in protection against severe disease. The second arm of the adaptive immunity is through antibodies. So antibodies are secreted and produced by our body, and it acts before the infection takes place. So according to the function of the antibody, it can be categorized into binding antibody and neutralizing antibody. So binding antibodies are antibodies that binds to the virus but may not interfere with its infectivity. And neutralizing antibodies binds to the virus and interferes with the virus ability to infect a cell. So as shown in this image here, when there is a presence of the neutralizing antibody, which is shown with the red schematic here, it can bind to the spike protein of the virus, thus prevent it from interacting with the ACE2 receptor. So as an end result, the virus is inhibited from entering the cells. So recognizing the important role of neutralizing antibody in preventing the virus from infecting the cells, several guidelines has been issued from the FDA regarding the use of neutralizing antibody specifically for the development and licensure of vaccines to prevent COVID-19. So according to this guideline, the functional activity of immune responses should be evaluated to assess the vaccine effectiveness. So here shows the most recent fact sheet of the Moderna and Pfizer vaccine. So as shown here, the effectiveness of the booster dose of the Moderna vaccine was based on assessment of neutralizing antibody titer, and the same for the Pfizer vaccine. So how is the neutralizing antibody titer determined using conventional method? So this schematic here shows the mechanism of the how we can detect the neutralizing antibody using live cells and also live virus. As shown here, such methods involve the use of the live cells and also the virus. This method, the advantage is that it is gold standard in specificity. However, as I mentioned, it involves the use of the actual SARS-CoV-2 virus. It actually requires the very strict safety conditions, which is biosafety level 3. This is the image showing the PPE you will need to wear for entering such facility. So it can be quite logistically demanding. And also such assay is quite tedious and slow and usually takes 3 to 5 days to obtain the result. And also because of the complexity of this assay, it usually requires highly skilled operator. And also the assay is quite expensive and also not scalable and not very suitable for high throughput applications such as mass screening. And also because of the protocol and material differences, the results obtained are usually not comparable between different labs. So to address these technical difficulties, a surrogate virus neutralization test, abbreviated as the SVNT, is developed by Prof. Wang Nifa from Duke NUS. So as shown in this image here, which I have explained earlier on, so with the presence of the neutralizing antibody, the RBD region present on the spike protein will be prevented from binding to the ACE2 receptor. So the surrogate virus neutralization test use this principle, and instead of the live cells and virus, it uses purified RBD and also the host cell receptor ACE2 to mimic the virus host cell interaction in an ELISA plate well. So as shown in this image here, the RBD in the SVNT is conjugated with the HRP. So once it binds to the ACE2 receptor, there will be a colored signal. And for samples with neutralizing antibody, the neutralizing antibody will bind to the RBD region, thus form a complex with the RBD. Therefore, the RBD will be prevented from binding to the ACE2 receptor and showing a much reduced color signal. So this SVNT, compared to the conventional method, it is much more user-friendly and it can be used in a regular BSL-2 setting. And the assay is also fast, can be completed in 1 to 2 hours, and is also automatable with the use of ELISA automated machine, and thus is suitable for high throughput applications. And also because of the standard protocol we use, the between lab inconsistency is minimized. And this assay also offers very highly specific detection of the neutralizing antibody that can block the RBD ACE2 binding. And also the detection is not limited to certain isotypes such as the IgG or IgM. So for all the antibodies that have the function of block the RBD ACE2 binding, it can be detected using this SVNT assay. So how does the SVNT correlate with the conventional virus neutralization test? So as shown in this graph here, the correlation of the results obtained from PRNT, which is the gold standard cell-based virus neutralization test, and also CPAS, which is the SVNT. So we can see that the correlation have an R-squared value of 0.9446, and this is considered very high for any biological system. And as for other assays, for instance, like other commercial anti-SARS-CoV-2 serology assays, which are designed to detect the binding antibody, their correlation with the gold standard assay is very poor, with the R-squared value less than 0.5. So we also validated the sensitivity and specificity using the PRNT as a comparator. As shown in this graph here, we have for both the positive and negative percent agreement, we have 100% when we compare the SVNT with the gold standard PRNT. And also because of the small volume required for the blood sample, this assay can be performed with two different blood collection methods. So the first one is the conventional venous plasma, which is obtained with a venipuncture. And then it can also be used with finger prick, which is also acceptable, as shown here by this agreement study that we have shown 100% agreement comparing these two blood collection methods. So next, because of the high sensitivity and specificity of the CPAS SVNT, it is authorized by the FDA as the first test that detects neutralizing antibody from recent or prior SARS-CoV-2 infection. And since then, we have also successfully registered the CPAS SVNT for in vitro diagnostic use in more than 10 countries. So the SVNT is not only available as a research tool, it is also available for the general public to walk into a clinic and order these tests to evaluate how well they have responded to the vaccine they have taken. So next, I'm going to discuss how do we interpret the results from CPAS. So as shown by this formula here, the results of CPAS is expressed as a percentage inhibition. So the cutoff value is 30%. So for values that is larger than 30%, it is considered positive. And for those that are less than 30%, is negative, means that there's no detectable neutralizing antibodies. And alternatively, if you wish to express the result in antibody concentration, we also provide the neutralizing antibody standard for you to generate a standard curve as shown below, so that you can interpolate the antibody concentration for your samples based on this standard curve. So what we have as an additional tool is the international standard for neutralizing antibody. So this will allow for the calibration and also harmonization of serology assays detecting neutralizing antibodies. So this image shown here is the previous definition of the international unit for the Hep B disease. So it's shown that a protective level of antibody for protection against Hep B is 10 IU per liter. And also for SARS-CoV-2, the WHO has come up with this international standard that is contains the neutralizing antibody as shown here. And in particular, we also note that for binding antibody assays, the arbitrary unit is PAU per ml, while only for neutralizing antibody, we can express our result in IU per ml, which is international unit per ml. So because CPAS is one of the assay for detecting neutralizing antibody, we will be able to express the result in IU per ml. So next, I'm going to introduce the method to convert the percentage inhibition obtained from the SVNT to the IU per ml. So as recently published in Lancet Microbe, we have established this very consistent conversion between the percentage inhibition and IU per ml. So as shown in the averaged data here, the R-squared value is 0.977, meaning that based on the model developed in this paper, we can very reliably convert the percentage inhibition to IU per ml by using the formula that's developed in this paper. So next, I'm going to discuss how neutralizing antibody it can be used as a predictor of the protective immunity. First of all, I would like to share the quotes from several thought leaders in the field regarding the neutralizing antibody as the protective immunity biomarker. So the first quote is from Johns Hopkins University, stating that ultimately, neutralization assays are the gold standard for determining if a patient has effective antibodies and protective immunity against SARS-CoV-2. And also, Dr. Anthony Fauci, who is the chief medical advisor to the US president, have always been advocating that neutralizing antibodies are the gold standard for protective immunity. So next, I'm going to show some high-impact scientific publications that investigate the relationship of neutralizing antibody level with real-world protection. So as shown in this Nature Medicine paper, it has analyzed the mean neutralization level induced by the vaccination and its protective efficacy observed in real world. As shown in this graph here, we observe a very obvious trend indicating that the higher the amount of the neutralizing antibody, the higher the protective efficacy. And also based on the most recent Moderna phase 3 efficacy trial, we also see this very strong association of the neutralizing antibody titer with the vaccine efficacy data. And in this study, it was identified that the vaccine efficacy is estimated to be 45 to 60% for vaccine recipients without any detectable antibodies, and it can increase up to more than 98% for recipients with the highest neutralization titers. Next, I'm going to share a study that describes a COVID-19 outbreak in a facial vessel with a very high attack rate. So in this study, both the Abbott IgG assay and the CPAS SVNT assay is performed on the 6 crew members. As shown by the results here, we see that all the 6 crew members are positive of the Abbott IgG assay. However, only those who are positive for the CPAS assay are protected from reinfection. So again, this highlights that the presence of the neutralizing antibodies from prior infection was significantly associated with protection against reinfection. So next, I'm going to share some real-world data that establish the threshold of neutralizing antibody level as correlate of protection. As shown in this first graph table here, so this is a study performed in Israel. It is shown that for people who are protected from the vaccine breakthrough infection, their neutralization antibody level is 250 in IU per ml, which is equivalent to 60 to 78% for CPAS SVNT, while those who have a vaccine breakthrough infection, their neutralizing antibody level is significantly lower, which is around 100 IU per ml and 60% CPAS value. And then another study that is performed in Vietnam, it was identified that the amount of neutralizing antibody required to protect against the Delta variant is about 82% to 91%. So from this real-world data, we observed that when the variant of concern is Delta, the protection level of neutralizing antibody will also increase, and this is mainly due to the amino acid changes near the spike receptor binding domain that could result in the reduction of the neutralization titers for the variant of concerns. So according to this Nature paper, the neutralization of the wild type and variant viruses was investigated for those who are vaccinated with the Pfizer vaccine. So as shown in this graph here, the gray bar is the wild type and the colored bars are the variant of concerns. So we can see that the neutralization level has decreased for the variant of concerns, and the reduction is most significant for the Kappa variant, whereas for the other variants, the protection is still we can see is still quite high. And from the recent summary from the CDC, the neutralization loss can range from 0 for alpha to about 7-fold for beta, and the delta is about 1 to 5 to 2-fold reduction. So also for the for Omicron. So for Omicron, some of the preprint have also investigated how does the neutralization level loss when it comes to the Omicron variant. And the preliminary data shows that there is a 20 to 40 fold reduction for the Omicron variant. So this is kind of expected because we know that there's a large number of mutations on the receptor binding domain of the Omicron. So many of you are interested to know that whether the SVNT can be used for detecting neutralizing antibody against the VOCs? So the answer is yes. So as I mentioned earlier on, in our assay, we use the HRP conjugator RBD as the antigen. And for the CPAS kit, which is supplied, the RBD supplied in the CPAS kit is the wild type RBD. And we do provide reagents that can be used as a replacement of the wild type RBD so that you can use it for detecting neutralizing antibody against the VOCs. So next, I'm going to share some of the results that is obtained using the VOC RBD, using a VOC RBD for detection of variant-specific neutralizing antibody. So first of all, I would like to share a study from Singapore. So as shown in these results here, so for patients infected with the wild type, we observe that their neutralization for the wild type is the highest, and is a slight decrease for the alpha variant, and a very significant decrease for the beta. And for alpha, we also observe that the neutralization against the alpha and wild type is still high, but also there's a decrease for the beta. And we also see the results for patients infected with the beta variant, and as expected, their neutralization against the beta variant is the highest. So next, I'm going to show some studies that compared the results obtained using the wild type RBD and the Delta RBD. So according to this study from Singapore, we see a very strong correlation of the results from the wild type and the Delta RBD, meaning that for the same sample with a higher value for the wild type RBD, most likely the Delta RBD value is also positively correlated. And we also observe this reduction of about 0 to 30% as compared to the wild type. And this study from Brazil further confirmed the observation. So when we tested the Delta infected samples, we observe a very consistent trend that its neutralization against the Delta RBD is significantly higher for the neutralization against the wild type RBD. And this is vice versa for the wild type infected samples. So this set of data shows that using the VOC RBD and with the SVNT, we can differentiate the neutralization response for the different variants. So how about for Omicron? So according to the study from Singapore, so the patient sample is obtained from those who are vaccinated with the mRNA vaccine, including Pfizer and Moderna. So as shown in this graph here, we see that for people who haven't received the booster, their neutralization against the Omicron are all very low. And once they receive the third dose, which is the booster, there's a significant increase in the Omicron neutralization. However, we see that the results can vary a lot from person to person. And then here is a comparative study of how is the neutralizing antibody response against the wild type, Delta, and Omicron for samples that have received a booster vaccine. So we see that all of these samples have very high neutralization against wild type and Delta. And while some of them still have high neutralization against the Omicron, and a few of them have a much decreased level of the neutralization against Omicron. And also this study from US also investigated the comparison between the wild type, Delta, and Omicron neutralization response. So from this study, the patient cohort include those who are naturally infected and also those who have received a vaccine. And the vaccine for this study include the Pfizer, Moderna, and also the Johnson & Johnson vaccine. So from this study, we also see a very similar finding that when it comes to the Omicron variant, we observe a very significant reduction of the percentage neutralization. And this is the case for people who receive one vaccine dose and 2 vaccine dose, and also 3 vaccine dose. And those who are naturally infected also shows the similar trend. So next, many of you are wondering whether binding antibody assays are able to differentiate the variant of concerns. So from this comparative study, we observed that using the CPAS SVNT assay, we can differentiate the neutralizing antibody response against different variants. However, with the binding antibody assay, we did not observe any difference for different variants. So this indicates that when we evaluate the binding antibody after the vaccination, it may give a false signal that you still have a very high level of binding antibody, but in reality, the protection against the variant of concerns may have dropped significantly. So this image on the right is a very good mechanistic explanation of why neutralizing antibody assay can differentiate, but the binding antibody assay cannot differentiate. So this study is a comparative analysis with 4 monoclonal antibodies. So we can see from this graph here that all the 4 antibodies have very strong binding activities. However, we see that only 2 of them have very strong inhibition activity, and these 2 are not necessarily the strongest binders. So this again highlighted that the difference between binding antibody and neutralizing antibody. So next, I'm going to introduce how the SVNT can be used for the next generation vaccine development, which is the Pan-sarbecovirus vaccine development. So we know that the current vaccine are based on the wild-type antigens. So many of us are wondering, can the speed of variant-specific vaccine development keep up with the emergence of the new variant of concerns? Which means that by the time the Omicron vaccine is developed, most likely the damage caused by the Omicron could have already been happened. So it is of very urgent need to develop a variant-proof or pan-sarbecovirus vaccine that can not only protect us from the different variants, but can also potentially protect us from the next COVID virus that may now is still in the animal, but may have the potential to transmit into the human population. So for this work, I would like to introduce the recent work by the CPAS inventor, where they investigated 8 individuals who are recovered from the SARS-CoV-1, which is the SARS outbreak in 2003. And these 8 individuals have received 1 or 2 doses of the Pfizer vaccine. And as for the virus they have investigated, so this assay is carried out using the SVNT. So so the RBD protein is used as antigen for these viruses. So we can see here that the SARS-CoV-2 virus collate, including the wild type and also the alpha, beta, delta variants. And then also this study also investigated the SARS-CoV-1 clade. So of particular note, we see that there are 5 viruses, which is the pre-emergent sarbecoviruses, which are now still in the animal and may have the potential to transmit into the human population. So for these viruses, because now they are still in the animal, so the live virus is not available. So it is not possible to perform the assay using the conventional live virus-based method. So in this case, the SVNT assay can be a very powerful tool to allow for such a study. So this is the result from the study. So we can see that for people who are vaccinated with the Pfizer vaccine, they have a very high neutralization against the SARS-CoV-2 clade. However, their protection against the SARS-CoV-1 clade is very low. And then for those who are vaccinated, those 8 individuals who are SARS survivor, and later on vaccinated with the COVID vaccine, we can see they have a very high level and broad spectrum of neutralizing antibody. And this finding is of major interest because it means that by administrating the cross-correlate vaccine, there's a chance that we can achieve a Pan-sarbecovirus vaccine using a similar method. So next, I'm going to introduce the dynamics of the neutralizing antibody response and also the antibody longevity for people who are recovered from the COVID-19. So as shown here, there are 5 different profiles. So the first one is the negative, meaning that after they infected with the virus, they do not produce any viable neutralizing antibody. And also the second is rapid waning, meaning that they have an initial high level but decrease very quickly. And also the slow waning and persistent group. So this study shows that the neutralizing antibody response dynamics in the recovered patients vary greatly, and the prediction can only be accurately determined at the individual level. So next, how about those who have received the mRNA vaccine? So this study here also shows a very similar trend of antibody waning over the time. And as we can see from these results, we also see a very huge variability for different individuals. And this study using the SVNT analysis also shows a very similar finding. So how about those who have received the inactivated vaccine? So here I would like to share a study from Hong Kong and another study from Singapore. So according to this Hong Kong study, for people who have received 1 dose of the Sinovac vaccine, most of them are still negative. And when they receive the 2nd dose, there's a significant increase to about the similar level for 1 dose of the Pfizer vaccine. So the study from Singapore also reported very similar finding. And to our surprise, although these 2 studies are performed in 2 different regions, the results reported are very similar to each other. As shown here, 1 dose of Sinovac for the Singapore study is 16% and 15.6% for the Hong Kong study. And the values for the 2 doses of Sinovac, 1 dose Pfizer and 2 dose Pfizer are also very close to each other. So this highlights that we can achieve very highly concordant CPAS results for 2 different regions. And of course, that is performed using different patient samples. And besides this, we also see this huge variability for different individuals, especially for those who received the inactivated vaccine. It can vary from about 90% all the way to negative. So this highlights the importance to adopt a neutralizing antibody-guided booster shot regimen. So as shown in this graph here, it is actually helpful that a mass screening of the neutralizing antibody can be performed among the vaccinated individuals and identify those who have not responded well to the vaccine, meaning that those with a negative CPAS or a very low level of the CPAS can actually be identified and selected for the booster dose to minimize the vaccine breakthrough infection. So this can be especially useful for countries that have that may not have enough supply of the booster dose for everyone in the country. So next, I would like to show a study that is performed by the WHO comparing the CPAS method and the PRNT method. So we can see here that for the PRNT, the blue and red represent the results from different labs. And we see that the results obtained from different labs can vary a lot for all the 6 samples investigated. However, for CPAS, we see that the results obtained between different labs are very close to each other. So this again highlighted the consistency of between lab of CPAS value obtained in different labs. So in conclusion, we know that only a fraction of antibody produced are neutralizing antibodies, and the presence of the binding antibody or IgG or IgM antibody does not mean that functional neutralizing antibody are present. And also we have shown a lot of evidence proving that neutralizing antibody is a good biomarker for predicting protective immunity. And also it is known that the neutralizing antibody level wanes over time, and the dynamics can be highly variable between different individuals. And also the SVNT can be the ideal tool for neutralizing antibody testing against variants of concerns, as well as those pre-emergent cervicoviruses. And lastly, we have shown very highly concordant CPAS SVNT results across different labs and different cohorts. So the application of the CPAS at SVNT can be summarized as the following 3 applications, including study vaccine efficacy and identify who and when need a booster shot, and also for the next generation vaccine development. So before I end my presentation, I would like to introduce some additional tools to accelerate your Omicron research. So at GenScript, we provide these recombinant proteins, including the spike protein and also the spike RBD protein and the RBD HRP, which can be used with the SVNT, as well as the M protein for the Omicron. And besides that, we also provide the antibodies, including nucleocapsid antibody and also neutralizing antibody standard that is proven to neutralize the Omicron. And also we provide the plasmids encoding the spike protein, encoding for the spike protein, as well as the peptide tool for the SARS-CoV-2 Omicron, as well as the pseudovirus itself, also the pseudovirus neutralization assay kit. On top of that, we also provide a customized service, including the oligo and mutagenesis, recombinant protein production, and also customers cell line. So if you require any of the customized service, you may get in touch with us. So I would like to thank you for your attention, and I'm happy to take any question you have.
All right, thank you so much, Dr. Chengxun, for your wonderful presentations. So right now, we will accept some questions from the attendees. So for all the attendees, you can ask any questions in the Q&A box, which is already highlighted now. Okay, Dr. Chengxun, here is the first questions for you. So the first question is, I have seen this CPAS test from GenScript website. So is there any difference between CPAS and SVNT?
Thank you for this question. So the SVNT is the technology behind CPAS. So it refers to the same product, while the CPAS is the trade name for the SVNT. So it refers to the same product, yeah.
Okay, thank you. So, Dr. Chengxun, so do you foresee this SVNT technique or the CPAS for a clinical use, or it will mainly be used in the research?
Thank you for this question. So right now, the SVNT technology has been widely used in research setting for evaluating especially for clinical trials to evaluate vaccine efficacy. And also it has been used a lot in the clinical setting. For instance, in Singapore, it is available as a serology test for people who can actually walk into the clinic and order such tests. So yes, it can be used for both setting, and it is valuable that it's a valuable information for the general public after they receive the vaccination. They can also test for the CPAS to know their neutralizing antibody.
Okay, so here comes our next questions that follow the previous one. So how long does the CPAS antibody level last after a single test? And since we are living with COVID, right? So does it mean everyone should take the regular CPAS testing over the years?
So this is a very good question. So as I mentioned earlier on the neutralizing antibody longevity, so we see that the longevity really differs from person to person. So we see that for people who the decreasing is very fast, and got people who have a very slow speed of the decreasing. And also the luckiest group of people, they have actually have a very persistent level of neutralizing antibody over the course of 6 months. So yes, the CPAS neutralizing antibody level only represent the time that you take the test. So after a period of time, it is recommended that you take another test. So usually this period of time can be about 1 month or 2 months.
Okay, thank you so much, Dr. Chengxun. Yes. So there's a next question here. So does the SVNT assays detect activity against all the current X protein variants?
So this is a very good question. So as I mentioned earlier on, so okay, so this is the answer to your question. So in the CPAS kit itself, the RBD provided is the wild type RBD. So it detects the neutralizing antibody specific to the wild type. And if you wish to detect the variant specific antibody, you can purchase these reagents, which are encoding for the variant RBD, and use it as a replacement for the wild type, so that you can evaluate what is the neutralizing antibody level specific to certain variant.
Yes, thank you. So our next questions will be, is CPAS the only neutralizing antibodies test on cells in global market, or is there any other company also selling the similar products, and how can CPAS differentiate from the other products?
Thank you very much. So CPAS, when we talk about SVNT, there are also other companies that sell very similar products, but our one is the CPAS is the first test that is authorized by FDA, and we are also have been adopted in both research settings and clinical settings very early on. So we do have a very strong publication list that you can refer to. And also besides this, we also have different products that can be used together with the SVNT, including the ones that I just mentioned for detecting of the variant-specific neutralizing antibody. And also we have a very important advantage is that we have this established correlation between the percentage inhibition and IU per ml, so that once you obtain the result using the CPAS SVNT, you can simply use a formula to convert the result into IU per ml, which is very valuable if you wish to compare your results with other literature. And besides this, we also have a neutralizing antibody standard that you can use to generate standard curve. So I would say that we are compared with other existing SVNT that is selling in the market. So majority of them are for research use only, but the CPAS is clinically validated, and it is a much more mature technology as compared to the other similar products sold in the market. Yeah.
Yes, thank you, Dr. Chengxun. So I guess that was the last questions that we have from the attendees. So thank you so much for your wonderful presentations for today. And thank you for all the attendees who, there's another questions coming. So wait, let me check this. So is there correlation between the results from CPAS SVNT and CPAS for Omicron variants in vaccinated participants?
So this is a very good question as well. So as I mentioned, that we do see a correlation of the CPAS wild type and also the Delta with the Delta RBD. But for Omicron, we have noticed a much more higher variability, meaning that meaning that for people who already have a high neutralization level against the wild type, they may still have a very low Omicron neutralization. So right now we are still there are still ongoing studies to evaluate the relationship between the wild type neutralizing antibody and the Omicron. So we will be sharing if we have any developments on that. But right now, we do not see a very strong correlation between these two.
Okay. So is all these correlation data on the vaccinated participants or non-vaccinated?
The correlation data, can I clarify, do you mean the correlation between the wild type RBD and the Delta RBD?
Yes.
Oh, so this correlation data comes from vaccinated individuals in Singapore.
Okay, so is there any correlations with the Delta variant?
Yes. So as I show in one of my slides, it is shown that the R-square value is about 0.8. That means that for people with a high CPAS value for the wild type, their protection against the Delta is also usually higher.
Okay, yeah. So, yeah, thank you so much. Let me see if there is any other questions coming from our attendees. Okay, I guess that should be it. So, really, thank you so much, Dr. Chengxun, for your presentations. And to all the attendees, we hope these presentations will be helpful and beneficial for your work. If there's any questions, please feel free to check out our website, www.genscript.com for more information. Thank you, and we'll see you next time.
Thank you, everyone. Have a nice day.